And so now I get the pleasure to introduce our keynote speaker. John Marioni, I have here his bio. Where is he? So John is the global head of Computational Sciences Center of Excellence at Genentech, and specifically the research and early development program called GRED. And in this role, he oversees computational scientists, both from Genentech and Roche, ensuring that all future AI projects serve the breadth of Genentech's research and early development activities. He's a member of the leadership team, reflecting the importance of computation, software, and quantitative approaches for research, drug discovery, and development. Prior to joining Genentech, he was the head of research at the European Bioinformatics Institute, part of the European Molecular Biology Lab in Cambridge, UK. As head of research, he was responsible for 16 research groups in diverse areas of computational biology. His work has been recognized internationally. He is a member of the organizing committee of the Human Tumor Atlas and was elected member of EMBO, and in 2022, was elected fellow of the UK's Academy of Medical Sciences. His lab has made particularly significant contributions in the area of functional genomics, including developing some of the most widely used approaches for the analysis of single-cell genomic data. In addition, with collaborators, his group has made important contributions to the understanding of cell fate decisions in the context of early development in the immune system and in disease, including cancer. A statistician by training, he obtained his PhD in computational biology at the University of Cambridge and was a postdoc in the Department of Human Genetics at the University of Chicago. So it truly is a pleasure to welcome John. Thank you so much.
Thank you very much, Sylvia, for that really kind introduction. I think when people go over your career like that, it makes you think, gosh, I'm old. It's kind of like, oh dear. No, but it's really kind. And thank you all for the planning. Thank you to the planning committee for inviting me here. It's always a pleasure to have the opportunity to speak to trainees. The ability to have to talk, to work with the next generation, who are the people that are going to have the bright ideas for the future, that are going to drive this field over the next 10, 20, 30, 40 years is always inspiring. One of the things I miss most from having an academic lab was that ability to mentor PhD students, that ability to work with people at that very early stage of career. One of them, Emma, in the audience here, was joined with me and Sarah Teichman, now with Jonathan Pritchard. That ability to have that interaction, that freshness of ideas, it's truly inspirational. So really great to see so many students in the audience today. I look forward to chatting to you hopefully throughout the course of the day as well. It's a great event and it's really wonderful to see this established here. So today I'm going to focus mainly on, of course, as Sylvia's introduction alluded to, the work that we're doing in Genentech in the context of drug discovery, and I'll talk to you a lot about this, particularly in the context of target identification for qualification, some of the foundation models we're developing there, how we're thinking about identifying targets. So a lot of work in the functional genomics realm that was also mentioned in the introduction. But before we get there, I thought I'd give just a little, very short background about my own personal research, how I came to Genentech, and why I made that transition from academia to industry. So as Sylvia mentioned, I was a postdoc in Chicago in human genetics, actually in the same lab as Barbara Engelhardt at the time. So she and I had a lot of interactions when we were both rather younger in 2008 to 2010, which is a lot of fun. And what I worked on as a postdoc was methods for bulk RNA sequencing data. So at the time, this was the state of the art. We were very happy with 32 base pair, 25 base pair reads. If you got 1 million reads in an experiment from a whole flow cell, that was a tremendous outcome. And what this figure here shows is work that was actually done with Jonathan and Yoav Gilad, looking at selection on gene expression levels across primates. So what you see on the y-axis there is just the phylogeny based upon the sequence, and on the x-axis is showing the structure of a particular gene, and at the bottom is a little phylogeny of how the expression level of that particular gene has changed. On the right-hand side or on the left-hand side, is what you would get if you just put the phylogenetic tree based upon the genome sequence. And so by doing this, we were interested in studying genes that were under particular selection and then linking that to environmental pressures. And we did this in liver samples across these species, primarily of primates. And the reason we did it in the liver was because it's pretty homogeneous. 70% of the cells are hepatocytes, and you're not going to be confounded with heterogeneity of cell types. But of course, we actually want to understand that heterogeneity. And so when I started my lab in Cambridge, that motivated me to think about how I could get into what was then the quite young field of single-cell genomics, in particular single-cell transcriptomics, where instead of having those bulk average measures across millions of cells, you were able to profile single cells. And over the 12 years or so that I was in Cambridge, that was really the focus of the group, thinking about methods for these types of data, how we would control for batch effects, how we could deal with confounded factors, and then applying those methods in different contexts, particularly, as I'll talk about very briefly, in embryogenesis. And so, together with a bunch of fantastic colleagues, mainly in the UK, but some actually in the US, in Long Kai down at Caltech at the time, we took single-cell approaches and applied them in the context of early gastrulation, early embryogenesis. And this is a system that's especially amenable to single-cell omics because you've got a small number of cells, they're extremely heterogeneous in their identity, and so averaging across all the cells in the embryo is just good to dilute all the signal. And so we applied a variety of single-cell omics approaches, not just RNA, but functional genomics and multi-omics data to profile cells from these embryos. This was work with Bertie Gutkins and Wolf Reich, so just basically throughout this time course, and as you are all no doubt familiar with, you could take these data, these single-cell RNA sequencing data sets. You can, after a lot of pre-processing, which now agents would be able to do a lot of it for you, but at the time was rather more laborious, you were able to take that high-dimensional expression matrix and project it after some filtering down into 2 dimensions. And this is just a UMAP representation of the data. Each point here corresponds to a cell. And if you're interested, you can begin to look at particular trajectories. You've got this little blood wing at the top. You've got the endoderm down at the bottom and the intercalation of the extra embryonic cells into the embryo proper to form the gut tube in population number 8 down at the bottom. So there's a lot you can do with this. You can look at build these trajectories, construct them across pseudotime, examine how individual gene expression changes along this process, begin to understand the regulatory networks. As well as doing descriptive work, you can also functionally interrogate this. You can use traditional knockouts and just compare the wild type to the mutant cells. We also worked on chimeras where we had we injected cells into blastocysts, so we had a mix of wild type and mutant cells, and by labeling the cells differently, you were able then to distinguish them, and that allowed you to understand whether the mutant cells, where you typically knocked out a gene, here TAL1 in the context of blood development, you were able to understand their function without disrupting the development of the embryo proper, because the risk of doing this in a total knockout is that the embryo it's lethal for the embryo and it dies. And then, and this is just a little nod to Emma's work, we also thought about some of the methodological work here. So this was Milo, so this is clustering-free testing for differential abundance, where instead of having to cluster the cells and then asking whether within a particular cluster we have different cells from conditions A and B, we're instead able to do this in a more agnostic way, identifying index cells along the graph of single cells, building neighborhoods, and then within each of those neighborhoods, asking whether we have differential abundance. More recently, we continue to work on some problems in early development. So my last PhD student at EMBL, Valentina Lorenzi, worked on the molecular mechanisms that drive human developmental organ development, in particular how the male and the female reproductive organs distinguish from one another. It's a really absolutely fascinating process that I'm happy to talk about afterwards. It's really beautiful, but not the purpose of today's talk. And then for the more methodological side, something that I'm very keen on is analysis approaches that don't require joint embedding. I think one of the big problems in the single cell field is that very often we start by creating a joint embedding of everything. We want to put it in the same space. The trouble is by doing that, you lose subtle signals, for example, related to sex, genetics, age. That joint embedding just loses signal that you care about. So I'm thinking methodologically about more meta-analyses like approaches where you're able to bring together neighborhoods from different studies without actually putting them on the same embedding. And by so doing, you could then do meta-analysis or just compute summary statistics across those to bring the data together. And I think that we've, as a field, the single cell field, rather obsessed about this joint embedding analysis. And I think that that's probably not scalable, and I'm not even sure it's the right thing to do if you're interested in subtle signal. For example, the start of disease or signal associated with some subtle feature that you care about, like genetic signal. And then at Genentech, and this middle work and the right-hand work is work that I'm doing at Genentech with some of my postdocs there, thinking about high-throughput perturbation screens. We have an amazing opportunity, and I'll talk a little bit about this in the context of our pipeline, to think about how we perturb systems. It comes with a lot of challenges. You could do this in a spatial context now. You can do it using optical pooled screens. You could use it using classical perturb-seq. There's a lot of thinking about how to analyze that data properly so that we can, in time, hopefully, be able to build models that will predict the impact of an unseen perturbation in an unseen context. That's ultimately what we'd like to do, because if we can do that, then we can begin to do a lot of biology in silico and design the next set of experiments in an intelligent way. So that's a very brief overview of 12 years plus of research. And I hope you can tell that I really enjoyed it. I really enjoyed doing this. It was something that I found very motivating, that ability to work with people, the collaborative nature of the work that we did in academia. Why make the transition and why move from the UK to California other than for the weather? Though today it's beautifully reminding me of a typical spring day in the UK, I have to say. It's kind of gives a certain sense of nostalgia when you look out the window here. So I think there were a number of reasons. I think that around the start of the decade, three things came together that were especially opportune. There was the ability to really generate data at massive scale. We'd been able to do it reasonably well up until that point, but the ability to really do it at very large scale and to perturb systems really came together around the start of the 2020s. Obviously, there were big advances in some of the underlying architectures, transformer architectures, that enabled us to do a lot more on the AI side. And then third was the compute, right? The ability to actually train these models, to have enough compute to train them so that they could be as powerful as perhaps we theoretically thought they might be, but we weren't able to tell because we didn't have the compute to effectively and efficiently train them. And all those three things came together, and it allowed us to think about applying them to what is, as Sylvia alluded to, one of the biggest challenges in the field, and that is going from a target to a drug. This is, and I think I underestimated this when I made the transition, this is really hard. It's both really hard and also really slow. And I think in academia, you're used to kind of that gratification after 12 to 18 months often in a project. You begin to see the germ of a paper and you begin to know, has my idea worked or not? Here, this is about 8 years, if you're lucky, from target identification through to a medicine, and it can fail at any point, including in very expensive phase 3 trials. And at the moment, you have a success rate of probably 1% from things that enter the portfolio to things that actually become a medicine that you start to manufacture. That's extremely expensive, it's extremely slow. And so what we have the opportunity with AI and computation to do is to not make this 100%, that's just too optimistic. But even if you were to increase that from 1% to 2%, that's already an enormous leap forward in terms of how you're able to think about this process. So we have the opportunity to get better targets, better drugs, to be more efficient in terms of content generation, in terms of filing, to reduce these timelines, and to have better targets coming in. And that's what we hope computation and AI can do. And that was the reason that I moved, because I thought that the time was right to actually try and tackle these problems using large-scale computational approaches. It's quite obvious, hopefully this doesn't give anybody a look at this, I realize now it's rather large and a little bit mesmerizing. It's quite obvious why this is a really hard problem. If you think about it, we've got such an enormously complex organism that we're dealing with. We have trillions of cells, thousands of genes, they can interact in lots and lots of complex ways. So if we want to be able to get to a particular target, it's going to be really hard. And even when you get to that target, you've got heterogeneity across individuals. You've then got to design a molecule to it. There are, trillions, 10 to the 32 possible antibody sequences that you could design, even more small molecule sequences that you could design. So not only getting to a target is hard, but actually then designing and understanding whether you can design a molecule that can have the right effect upon that target is also difficult. There's no way we could experimentally, even in our wildest dreams, think about synthesizing all of these compounds and testing them. So we've got to explore this computationally. And so this is what AI allows you to do, in particular generative AI. It allows you to explore, based upon representative data, different parts of this space. And it's nothing new in some sense, because technology has always driven science. Over the last, 40 years, there's just a little timeline here of some of the major advances. You can see that very often biological discovery, chemical discovery is driven by technology, whether that be about the cloning of particular genes and cloning technologies, through to some of the early advances in deep learning, functional genomics technologies, RNA sequencing around 2010 and on. And over the last few years, we now can begin to see that AI begins to play into this as well. So it's the next technology that's going to be transformative. Now, arguably, it might be much more transformative and disruptive than some of these technologies, not just in biology, but across society. And one of the things I was talking about with Karen before is that societal impact and how it impacts the work that we are all going to be doing, but in particular for that next generation of trainees, the work that you're going to be doing and how you think about setting up your lab, how you exploit AI to really utilize it best. That's going to be critically important to embracing the future, which is why it's so great that Stanford has this program here and we have the ability to talk about it today. But in the context of what we're doing in the pharma industry, it's another of those technologies that's going to enable us to push these things forward. And in Genentech, you've heard this phrase a lot, and it's probably percolated through the entire industry by this point. We do this using a paradigm called lab in the loop, which is really actually conceptually quite straightforward. You start with data, large-scale data sets. You train a model. You then use that to generate some hypotheses. You test those hypotheses. That feeds back into the model. You do this iteratively, and over time, the model improves in performance, and then you're able to get better predictions and use that to either get better targets, better molecules, to design better trials. It's just reinforcement learning, but in a paradigm of a lab. And many of the challenges that we see are on the computational side, but I would say that, and it's worth never forgetting this, there's a lot of just how you operationalize some of this that's hard. Just people are used to working in particular ways, and then you change how people work, because this does require us to think about how we change the way we work. We shouldn't underestimate those challenges here as well. Some of the more mundane operational aspects here actually are, in reality, a little bit what can hinder our ability to accelerate and take this forward as fast as we might like. Today, I'm going to focus primarily on the left-hand column here, how we're thinking about using AI and ML in the context of target discovery and assessment. I'm going to give you a number of different vignettes of approaches that we've developed and how we are using them in a number of different biological contexts. But before I get there, I just want to touch briefly on the two boxes in the middle, and that's about the drug identification. And this is, as we all know, perhaps the area where AI has had the biggest impact thus far in the early drug discovery space. Using AI models to predict molecule structure, protein structure, won the Nobel Prize a couple of years back. And I would say that almost everyone in the industry is now using AI in some way in order to better design molecules. And that's no different for us. So how do we think about this? Well, we think about this both for large and small molecules. This is just an illustrative example here. But basically you have you denoise voxel grid, so that means that you split this molecule into different little voxels here. You then add noise, and then you want to reconstruct that, and you want to move from that initial template sequence to a molecule that's going to have the desired properties. I'm not going to go into the details. It's really not the purpose of today's talk. There's a link to an archive paper if you're interested in this. What is important is that this can cut times by at least a quarter in terms of our ability to go from an initial sequence to a small molecule that can potentially enter the clinic. This is getting even better now as we begin to incorporate more and more information. That was really mainly sequence-based, but we can also think about how we evaluate fitness. We can mutate the molecules. We can filter for particular properties. I would say that filtering step and how you do it is actually very important. And by doing this, we can begin to cut the iterations from one set of molecules to the next by, say, 80% in the first instance, then by faster and faster and faster if we do this in a really well-organized and effective way. So this is a way that allows us to go from that large search space down to particular candidate molecules much faster. Similarly, in the antibody space, in the large molecule space here, we deploy a similar process as a diffusion model where you start with random noise, then you begin to denoise it. You can see some of the intermediate steps in the middle here. And then after training on enough data, you're able to begin to generate sequences that look like the kind of candidate that you're after. So this is a particular example here where you see a particular loop in the sequence. So we've been able to deploy this quite effectively. And again, over the last few years, we've been able to add additional pieces of information to the data. So, for example, testing for immunogenicity, and we're able to do that in silico, and that allows us to optimize how quickly we're able to get to good molecules. And then finally, we are able to roll this into an agent-based approach so that this is accessible to people in the lab. And this is something that I'm sure James will talk about more this afternoon. So no great surprises here in what we're doing. Okay. But what I really want to focus on, that was just a snapshot, I'm happy to talk about this afterwards, is this target discovery and assessment. And I already mentioned why this is hard, and I think it's illustrated again here. Biology is multi-scale. Our measurements are extremely heterogeneous. Some of the assays that capture patient context best lack causality, so we're not able to go and infer, just say, from single-cell RNA-seq or methylation what's causal. Others give us intervention, but they are further from patient context. If you think about, say, cell line models or organoid models, they're useful, of course, but it's one step removed from a patient. So you're able to summarize a patient well with some data sets, but the perturbations that you're able to do, even with patient-derived organoids, they're still not the same as actually the tissue context. The best organoids don't capture that especially well. And finally, analyses are still typically done in a stepwise way. So we have error propagation at every step, and we end up traversing this massive landscape without really accounting very well for the complexity. We kind of deploy very often a plug-and-play-like approach where we'll start somewhere, we'll do some analysis, there's error in that, but we kind of ignore it, and then we go on and on and on. What we hope, and I think this is a hope not just for us in Genentech, but across the field, is that foundation models and the lab-in-the-loop-like approach, the reinforcement learning approach that I described earlier, can begin to help us to address this. Foundation models change this by learning from large integrated data sets and hopefully generalized to allow us to answer many questions in parallel with better accuracy and translation to human predictions in time. And then through that RL phase in the lab-in-the-loop, we can begin to steer the next experiment, close that loop between data generation and modeling, so that we're able to take account of all of the data in a holistic way, and we don't have this stepwise process with lots of maybe inefficient interventions. And this isn't just a figment of the imagination. At the moment, we can begin to see that foundation models in a biology context are beginning to become meaningful. They're not there yet in all areas, but they are beginning to become meaningful. But they're meaningful under a number of different conditions. I think that this figure, which many of you again will have seen, illustrates the context where they begin to become reasonable. And it's two things that I mentioned before. It's the data size and it's the GPUs, basically the model performance, the scale of the model, if you think about it, that y-axis that way. And why this matters is that it emphasizes that the x-axis is as important as the y-axis. The ability to generate really large data sets in order to feed into these models really matters. And I would argue that perhaps more than the GPUs, is what's going to drive this forward in the next phase, that ability to input really heterogeneous and large data sets. It's perhaps there that we're going to see the biggest bang for buck. And what changes at the inflection point? Well, with enough data, the large models start learning hidden structured features that the simpler models can't. In vision, that's moving from raw pixels to semantic concepts, fur, tails in the context of these not very cute little doggies and catties. And in biology, these concepts look like cis-regulatory grammar, cell states, things that you're able to learn rather than just putting pairs of cells together on the basis of just the similarity of their transcriptome, but understanding that next layer, the grammar, the regulatory networks that underpin it. If we have enough data, then we can begin to build those models. We're not there yet, though. Literature language models and virtual biologists are helpful, but they've not solved all the problems. Why? Well, biology is measured, not written. Biological data are quantitative measurements of physical systems, and we shouldn't forget that. And scientific texts and papers are an abstraction of the data. So just taking the papers, using an LLM to ingest them, that's useful. It gives you a lot of information, but it can only get you so far. And in particular, those models can't learn effectively in areas where there are few or no measurements. It's just going to, the generalizability of these models is going to be tough. And again, progress in biology follows new measurements. And so we've got to think about how those can drive what we're doing. Okay. So I was going to talk about that, but I'm going to move just straight on to 4 short vignettes of work that we're doing. And I'm going to start with genomic foundation models and going from DNA to mechanisms to functions. Okay. So genomic foundation models, or DNA language models, as they're sometimes referred to in the literature, are trained on large collections of genomic sequences paired with functional genomic measurements, chromatin accessibility, histone modifications, gene expression. And the goal is fairly simple. We want to learn from the DNA sequence, can we predict what the functional readout will be? So from a DNA sequence, it might be in the promoter region, might be a bunch of transcription factors upstream of a gene, can we predict the expression of that gene? They could be trained in different ways, supervised, unsupervised, semi-supervised, but at the core, the training process forces the model to learn patterns in DNA sequence that explain regulatory activity. In other words, they begin to learn the regulatory grammar of the genome. And this is a field that I would say in the biology foundation models area, this is probably the most mature at the moment. And we can see when we interrogate these models that they capture elements of the cis-regulatory grammar of DNA, for example, transcription factor binding motifs and their combinations that drive regulatory activity. And work from a number of colleagues, including Anshul here, has really driven this field over the last few years. In terms of what we've done internally, we've taken some of the principles from these models and thought about how we can model sequence to function, but at a cell type resolution. So this is the first meta-cell resolution sequence to function model, and this was trained on 7,000 cell states. And the goal here is in each of these different cell states, can we predict from the sequence the expression in that particular cellular context? So we're able to take advantage of these large atlases of single cell RNA sequencing data, and then take some of the models that have already begun to predict sequence to function and effectively fine-tune and retrain them on this larger corpus so that we're able for any particular cell context, if we give the regulatory grammar and its cell type X, can we predict the expression of a particular gene, the activity of a particular transcription factor. So it takes this input, segments of DNA in cis, and then predicts those expression levels. And as I say, we could do this across these 7,000 cell states. And there are many downstream workflows and tasks that follow from this. So we could think about, can we use the model in reverse to predict promoters with specific gene expression that will have for specific Treg gene expression? We can think about this in the context of variant effect prediction. What's the functional consequence of genetic variation? One real example that we have was thinking about designing genomic DNA or RNA sequences. So these applications span areas such as gene and cell therapy as well as vaccine design. So here we're trying to design promoters that drive expression of a rescue gene only in a specific subset of cells, for instance in neurons, but not in other cell types of the brain. So we can think about how we can design sequences where basically we can predict in the model that they're going to drive expression of a gene in the brain, but not in other cell types. So they're kind of logic models where it's here, but not there. Another way that we could think about this is patient-specific RNA sequences based on tumor profile. So here, this is about in the context of our mRNA cancer neoantigen vaccine program, and we want robust UTRs that will work across different payloads, and we also want patient-specific mRNAs that will translate with variable efficiency. So here, what we have is the sequence. It could be the UTR sequence, and we want to relate that to translational efficiency. And what I'm showing here is on the y-axis, I'm showing the experimental data, and on the x-axis is the predicted translational efficiency from one of these models. And what you can see is that although it's not perfect, it's actually pretty good. So it allows you to think about how I might design an optimal UTR that's going to give me the translational efficiency that I want on the basis of a particular sequence. So this is external data, 3,200 sequences that have these different UTRs. So it's kind of an independent data set that the model was not trained on. It's a decent correlation, 0.65. There's more work to do to push this forward, but already you could begin to see the potential of these methods for designing particular sequences that are going to be suitable in particular patient contexts here to modify translational efficiency. A second area, what I'm just going to talk briefly about where we apply these models, is in understanding biology at a grander scale, something that we call pathobiology maps, but in essence, you could just think of this as taking all of the data we have to try and best understand a disease. So these are some of the different diseases we study. On the left are some of the different data types, so from human genetics, functional genomics assays, et cetera, and we want to be able, for a particular variant of interest, it might be one that's highlighted in a genome-wide association study, to understand what is that a good target or not. That's basically what we want to do, and we would like to be able to rank those effectively. So of course, there's a lot of public data on this. There's the GWAS studies, there are functional genomics genes, EQTLs, etc. We also want to but there are many loci that we don't actually have information about. We don't actually know what they're going to do. We don't have good causal information. So this is one example from IBD. So here there are at least 300 loci from GWAS association studies, 1,600 candidate causal genes. So using literature, slow process, we're able to go through and link, say, 200 of these genes with a particular, or variants with 200 of these genes. So we're able to tie a particular variant to 200 genes and say, okay, this is influencing that, and we understand the context. But there are several hundred loci that remain unresolved. And what we can then do here, using the Decima model, is to in silico perturb the sequence. So we know these variants, we can in silico perturb it, we can begin to understand what's the effect of that perturbation in silico and predict what gene expression is going to change as a result of this perturbation. So we take advantage of this model trained on a wide corpus of data in order to understand the impact of an individual variant. And that allowed us to go from the to basically assign identification to the causal gene for about 120 additional genes. So we were able to go from that initial set and find another 120 genes that were, or to link variants to another 120 genes, which I think is rather nice. So moving forward, we want these maps to improve in quality. It's also an area where I think that as it's quite mature, we can think about how we learn from existing models, how we can even self-train them over time using agentic approaches. But at the moment, this is kind of our state of the art, and I think it compares pretty well with what else is going on in the industry, the Decima model. So that's one little vignette. A second vignette, and it's something that, again, Sylvia alluded to, were these large-scale single-cell foundation models. So here, over the last decade, we've generated hundreds of millions of single cells using single-cell RNA sequencing, and it's now at the scale where we can actually train foundation models. So to address this problem, Graham Heimberg, who's in Sarah Mostafavi's department in my organization, he and his colleagues developed something called Similarity, which maybe some of you have used. This is a single-cell foundation model that jointly leverages gene expression profiles and text-based cell annotations to learn a unified representation of cell states. So the model was trained on over 100 million single cells, and I think it represents one of the more powerful approaches for interrogating single-cell RNA sequencing data that's out there today. It blends unsupervised representation learning and supervised metric learning, so it's a combination of supervised and unsupervised training here. So the supervised part is that you create a supervised triplet loss function. Basically, you consider 3 cells, 2 of which are similar to one another, that we know because they have the same cell state, one of which is in a different cell state, so that's the triplet that we create, and obviously we want to train so that we keep the 2 that are in the same cell state together. They separate from the one that's far apart, that's a different cell state. And there's an unsupervised mean square error reconstruction loss function, and this encourages the model to preserve variation for the input expression profiles and allows you to capture more subtle signals that would be lost if you only consider this representation, this triplet loss function here. So what you can do then is you take this is published work over the last couple of years, so in 2024, and then Max Gold extended this in 2025, and now I think that's actually even now published, it's not just on the archive. But what it allows you to do is to then interrogate these data to ask a variety of different questions of it. So one obvious one, and we use this all the time for this purposes, given this expression profile, where else do I have a cell that shows a similar profile? So we might be studying a particular gene, a particular cell type in a particular context. And we might have a drug that works really well there. What we're interested in is there another context where I have a cell type that shows a similar profile and I can then repurpose the drug? So is that clear? It's a relatively simple task, right? I've got a expression profile and just where else. It sounds really simple, but actually the ability to search that data set efficiently, that 100 million cells, is not trivial. And so what similarity allows you to do is to interrogate that and to find those other cell types. So this is just one example here where we have, we were interested in a particular target, and then we found another cell type on the right-hand side that actually showed fairly similar profiles in terms of the expression, and then we could think about repurposing. And we've actually applied this in a number of our internal programs as a way of thinking about how we repurpose some of our existing molecules. You can also think about this in other ways. So this is looking for bispecifics. So here we have pairs of cell surface receptors that are highly expressed in tumors, and we want to be able to design, we have molecules that can bind there, but we also want to ensure that the molecule doesn't bind in healthy cells. So what we want basically is the logical gate is we want gene 1 and gene 2, which are the expressed on the surface of the cell. We want them to be expressed only in that cell type. So what we're asking logically here is we want A and B to be expressed here, but not as a pair anywhere else. And so we can interrogate the similarity data set in order to ask that question. So you can then imagine asking a variety of other logical questions, logic-based questions about these data to take that foundation model and go from what is a very large universe to actually specific pairs of cell surface receptors that are specifically expressed in the type of interest and not expressed elsewhere. And again, this is something that we use, and I'm sure you'll understand that I can't go into details of what we find here, but it's a way for us prioritizing how we think about designing molecules in the context of oncology, for example. Okay. So a third vignette, perturbation foundation models. I think this is perhaps the area where there's been the biggest revolution in technology over the last few years. We started with rather messy perturb-seq technologies that were challenging for a number of reasons. We can now, those technologies have themselves improved, but in particular, the ability to scale this up using alternative readouts, in particular imaging, allows us to scale this. So optical pooled screens, where instead of reading out the transcriptome, you instead look at the morphology of the cell as a way of deciding the phenotype. So you start with a particular field of cells, it could be a cell line, you then treat that, you have a readout to know which guide is in which cell, and then you use the morphology of the cell as the way of knowing whether there's a phenotype or not. And combining that optical pooled screen with perturb-seq technology allows you to really scale this up because it's so much faster to generate large-scale OPS data and cheaper than it is to generate perturb-seq data in practice. So this is an area that I think has really moved forward, and as we go into the next few years, I think we're going to see more and more complex models, so thinking about organoid-based systems, in addition to just cell aggregates or combinations of cells, are going to give us more and more realistic phenotypes. And thinking about how we can then apply some of these Perturb-seq or optical pool screening technologies in those systems is going to be, I think, something that we and many of us are going to be applying in a variety of different contexts, both in drug discovery and in basic biology. Okay, so there are some models that are good at predicting perturbations, but we wanted to adapt some of these and apply them in our own context. So this is now a work of Russell Littman, who works with Dave Richmond, again in Sarah's team, and together they built a model called Presage. And this model combines prior knowledge about gene and cell states from the literature. Again, it's taking advantage of that literature and the data context in the same way that Similarity does. So you're kind of using prior knowledge as appropriate so that you do power the models with the limited perturbation data that's available at the moment. And this is actually an area where the literature and other information is really important because there just aren't that many large-scale Perturb-seq profiles out there at the moment. So if you really want to learn this effectively, I know that colleagues here in Stanford are working on that. But I mean, there's still relatively in the public domain, not so much. And interestingly, Russell found that 2 strategies were sufficient to produce informative gene embeddings across diverse knowledge sources. And here the goal is to basically put genes together that'll have similar effects on as a result of a perturbation. So he started using public data, things like Reactome, where we have known interactions between pairs of genes, and he computed node embeddings for all nodes with the nodes corresponding to genes. So simply just create an embedding from Reactome for a gene about pairs of genes that are related. And there are a variety of other databases, Geo, etc., that you could do similar work in. There are then Perturb-seq data, where you can apply PCA and extract principal components, scores corresponding to each gene's identity. So for the limited Perturb-seq data that exists, we can take that and we can say, well, given this perturbation, what was the effect upon the gene's expression profile? So we got the kind of the embedding space of a gene from the literature and also from Perturb-seq. And interestingly, just concatenating all of these different sources of information, so the embeddings of the genes in the Perturb-seq space, the limited Perturb-seq space, together with those literature information, actually created an embedding that was fairly similar to an unseen Perturb-seq map. So on the left is the knowledge source embeddings, where you just concatenated all these data, projected it into a little UMAP, and on the right is the Perturb-seq map, and this is for the, so this is where it's not seen the K562 cell line or any of the perturbations, but actually, although they're not entirely overlapping, which you wouldn't expect, the joint embedding isn't terrible, suggesting that maybe there's something that you can build on here. So given that start, what Russell did was that he took the embeddings for a given perturbed gene and then transformed them into a common latent space using source-specific mapping. So that's taking those variety of different embeddings, the different sources, pulling them into one so that I have in that space, just as I've got all my genes in a single common latent space. And then you compute a weighted average of the transformed gene embeddings, guided by an attention framework that combines the perturbation specific and the global weights. The global weights come from the literature. The local weights come from Perturb-seq information, where I have specific Perturb-seq data. And then he generates a perturbation profile for the perturbed genes by just having a very simple linear layer that transforms this average embedding for each gene into predicted log fold changes. So for each gene, I've got, you can think of its latent space as responsible to different perturbations. And then in that context, I want to see along that embedding, can I link that to a fold change? That's basically the idea. Does that kind of make sense to people? I've got an embedding of genes response to a perturbation, and I want to change that into a score to predict what the fold change would be. And then that allows you to predict the effect of an arbitrary perturbation in a given cellular context, because we got information of these genes in their different local contexts. We also have an estimate of what the fold change would be, and so then we can predict if I perturb this gene in this way, what would the cellular context be? It seems to work pretty well, actually. So on the left is the relative mean square error for a subset of genes that are selected at random, and what you can see here is that lower is better. So we want to have a lower mean square error in terms of our prediction. And across a variety of contexts that were held out, Presage, in combination with the small amount of perturb-seq data that's available, seems to do pretty well at predicting the effect of the perturbation, and also in terms of the directionality, which is shown on the right-hand side here. If you're interested in more details on this, there's this paper in the archive that I think is now in press, actually. So please go and do have a look at that. I think this is extremely powerful. It's early here. We're generating a lot of perturbation data in-house to support this, so perturb-seq data creating really large foundational data sets to support the training of these models. But the early results, combining the literature with the limited amount of perturb-seq data that's available already, do indicate that there's some promise here. So finally, you'll probably be glad to hear, and this brings me on to the final part of the talk, which is thinking about more clinical related data. So we've talked a lot about different types of foundation models, mainly in the context of target biology, so target qualification, improving the identification of a target. But of course, we want to bring this towards the patient as fast as we can. And here we have the ability to interrogate not only the sort of the functional genomics readouts that I've talked about before, but also clinical data, so electronic health records, imaging, x-rays, pathology, claims data, the whole lot. You could think about how we would bring these all together here. And there are, of course, many, many tasks that you could think of here about how you would design appropriate studies, how you would link the functional genomics data to the patient data. And I'm just going to talk about a single one of those. And that's actually kind of related to the previous problem that I spoke to. So here, the idea is that we've got text-based longitudinal patient data together with RNA readouts from that person. And we want to be able to link clinical trajectories with the underlying molecular state. So we have a large-scale data set of clinical data. We've linked the clinical records with the functional data. And we want to be able to then build a generative model of that so that we can ask questions such as, if we intervene in a particular way, what would be the change in the expression profile? And so some information about the model is shown on this slide. I'm not going to go into it, simply not time. But this is just an illustration. So here, what we have is real measures of gene expression on the y-axis. And on the x-axis, we have kind of a default condition in which the model's been trained in red. And then in condition B in green, and I apologize, I realize this is not good for colorblind people, so apologies. Condition A is on the left, B on the right for each panel. We ask the model what would happen if we added these additional pieces of information. So you can see we have TP53, there's an alteration, and then we give a bit more detail about what would happen in an altered case. So there's now a missense mutation. We give a particular insight about the clonal class of the particular cell or the cancer in this case. And then we can predict what the expression would be in this unseen state. And actually, we do pretty well. So it's not shown here, but we can predict this, and then we can go back and find a patient with those characteristics and see, do they actually have the change in expression profile we would expect given our model. So this works pretty well. It's still, I would say, very early. Here, our modeling is probably less mature in some of the other areas, but it gives a flavor of the sorts of questions that we'd like to be able to ask. So again, counterfactual questions. How does a specific intervention drive changes in gene expression and then phenotypic transitions and in the way that the disease develops? Okay, so with that, I'll close. I hope I've given you a flavor of some of what we're doing, how we're thinking about foundation models, especially in the context of target discovery in Genentech. What's exciting is that I do see this beginning to change our portfolio. I do see it being used, and I do see it having a positive effect. So this isn't just academic work, which of course is tremendously valuable, but it's actually really beginning to change our portfolio. We're beginning to use it all the time. These models are becoming increasingly powerful. The ability to predict the effect of a perturbation, the ability to mine really large-scale genomic data sets, to go from sequence to function, and over time to think about how we're doing it in the clinic. This combination of data, the model, this in time this iterative cycle so that we can really speed up this process and predict the next experiment, it's coming together, and it all matters because this ultimately is going to impact patients. We want to develop medicines that are going to impact the life of patients across a whole variety of different disease areas. And if we do this well, it's not that we're going to solve all of them, but we'll get to reasonable treatments faster. Thank you very much.
Hello, thanks for the talk. My question is, you talked about the drug discovery pipeline. Maybe 1% of drugs make it to market and they could fail anyway, and trying to boost those numbers. Do you find in practice or in your experience that this lab in a loop model is helping increase the numerator, like more targets that are making it, or like decreasing the denominator that you're filtering out more things earlier?
It's a great question. The honest answer is we don't quite know yet, because you only know if it succeeds after a successful phase 3, and still we're talking years. What we can see is that we are increasing the number of targets that enter. So I think that it was on one of the slides through the pathobiology maps, which are basically these foundation models. We have a 133% increase in targets entering our portfolio across a variety of different areas. So that's one thing that we can quantify. I think we hope that those targets are better and that they're going to get all the way through. The truth is, at the moment, we just don't have that information. And there are so many. So if I could wave my magic wand and get a particular data that would help, it would be safety, toxicity. It's just, when I was talking to students in Cambridge about this, when I went back a few months ago, I wish I'd paid more attention to pharmacology when I was in academia, because it really, 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 really matters. And I always thought it was, okay, whatever. But actually, it's so critical, the pharmacokinetics and the number of reasons that what look like a really promising candidate can fail when they get into an animal model, let alone a human model, are myriad. And we don't have enough data to predict that well. There's just not enough data out there. There's just not enough data either in our company or in others to really build good predictive safety models, especially at that early stage. So we have things that look promising. From our foundation models, we think they have the right function. We predict that hopefully their target safety will not be too bad, but until we do some of those experiments, it's really hard to know how many of them are going to translate through to that next step. So our hope is that we increase the probability of success and we have more. But I think until we get through this next phase and we enter early development with some of these candidates, it's difficult to know. I wouldn't be willing to say we've succeeded. I'm hopeful, but I would be lying if I said yes.
All right, thank you very much.
Hi, Peter Jackson at the School of Medicine. It's interesting to expand on the single cell analysis, and of course, it's in theory wonderful, and you can imagine finding communications between cells, ligand receptor interactions, which would be fantastic. There are some problems. Though one of the problems is that ligands or receptors, in particular, don't express very well. They don't need much message, and so you don't get those. And then also, there's an awful lot of steady state measurements, which really don't show much because they just show things at a resting state rather than things in a perturbed signaling state where you might see a lot more excursions that are meaningful for signaling. But I'm wondering now, at the scale you're working on, if you're getting enough data from different patients in different cancers, let's say, that would allow you to sort of start to see these variations more defined. Because we have a lot of students, for example, who are doing similar kinds of analysis with exactly the same tools, and they're doing very different tumors. And they're getting exactly the same pairings, which makes us worry that you're getting a sort of a generic answer. So,
yeah, I think that that's a valid concern. I think that one of the things that's going to help there is spatial data at scale. I think the dissociated data and inferring interactions, it's harder. Once you have spatially resolved data, you have that. If you make some assumptions about the area in which the interactions are likely to arise, it's not always the case, but it's likely to be somewhat local to the signaling cell, then I think that that allows us to build better predictions there. We've seen some evidence of that in early development, that you can predict those better, and even the subtle signals are reinforced when you have that additional X, Y, Z information. I think that in order to really dig into this more, probably the spatial data and integrating that into these models is going to be really important in understanding the spatial niche. So there's some really nice work at the moment about how you embed niches in a high dimensional space. So instead of embedding cells, I embed the local niche, which might just be like the X neighbors of a target cell. And then how do those local niches embed? And it's the combo of the spatial data and the omics data that might give the granularity that's needed to find that subtlety. Because otherwise, yeah, you're going to find not delta cyclic, whatever. It's going to be just kind of stuff that is not that deep. But I think the spatial will matter.
Thanks for the nice talk. I'm wondering if now many different companies basically create same data sets, use same models, will there be a convergence of the drug discovery process, and in 5 years everybody's launching the same molecules?
I doubt it, to be honest. I think, and why do I doubt it? Because over the last few years, people have had access to many of the same data sets externally. It's the prompts, the questions you ask ultimately, that are going to matter. We might have similar models. There might be differences in the architecture. There might be differences in the training. But the ideas that we have and the prompting, that's going to be a discriminator, and it's going to distinguish how we work. So I don't think so. Will we end up using similar models, or at least in principle similar models? Maybe, but it's the question. It's a competition.
Hi, I'm Benedict, postdoc in the biology department. I want to kind of follow up on the first question, but also in the question just now, asking if you kind of look back at all the candidates that Genentech had historically, the ones that were successful, the ones that ultimately weren't, would those be candidates that you would have picked out with these new methods also? Or is there like a different, is the diversity of the things you pick out with these newer methods smaller, larger, different, just a different class of targets?
Yeah.
So how does it kind of compare?
It's a great question. I think that I would say that it's harder and harder to find novel targets. A lot of the low-hanging fruit has passed. So the challenge is then more maybe, so the way I like to think about it, there's not a magic disease chromosome that we've not seen. There's not this unseen chromosome. It's more about interrogating these data and understanding them better in more subtle contexts, where we're going to be able to maybe target the medicines better, understand more about the heterogeneity of disease, and work out this subpopulation with this particular cell type functionally. That's where we can have an effective medicine. I think that maybe before there wasn't that understanding of the heterogeneity of disease. If you think about it, I mean, it's not that long since, say, Herceptin or Avastin in the context of breast cancers. Before that, it was very all-purpose treatment. So it's only really in the last 2 decades that you begin to, even in one of the best studied cancers, to have a little bit of specificity in terms of the treatments. I think what we'll see moving forward is an ability to understand a bit more about the specifics of a particular target in a specific context. Because that lower hanging fruit, that kind of broader medicine, that's probably in many cases been discovered. Because we understand enough about the basic mechanisms. So I think that yes, you would discover these, but so would almost any method. And we might have known about it before, we just didn't have a drug to it. But I think it's going to be that drilling down to that specificity where maybe we can hope to have a real impact.
Saurabh Shah from Memorial Sloan Kettering. John, always great to hear your thoughts. Thanks for a fascinating talk. So can you expound on what you think are the data blind spots that we currently have, let's say in oncology, and how do we fill those in the next decade, let's say?
the thing that worries maybe is the wrong word. So much of our insight is based around cancer cell lines still. And cancer cell lines are great and have been extremely insightful, but they're not the disease. And so the cancer tumor organoid models are, I don't know. I mean, you know more about this than me, Sarah, but I still think there's a bit of work to do there. And so I think the interesting question is, do we need better models of the disease, or do we need more modalities? Like maybe it's proteomics, spatial proteomics. A lot of people would say spatial proteomics is going to be tremendously useful. It's, I think, really interesting to think about, is it that, like more modalities or better models or both? And you could actually argue the same in the computational space. Do we need better models? Right? If you think about some of the underlying architectures that we have, they do, to some extent, treat cells as fixed, like chips. Some of the models. Cells move, they migrate, they die. These perturbation models might say, okay, I can predict what will happen to this cell in this environment. Actually, the cell's got to get to that environment. And it might divide. Some of its daughters might die in that process. The current models, the current high throughput foundation models, don't capture that. So the combination of some thinking about the underlying biology in the models, I think, is interesting. In particular, capturing actually what happens to cells rather than just treating them as inanimate, fixed things is one. And then it's this combo of what is going to be the most useful data. And for me, at the moment, that's one of the things that worries me most, because these are really expensive data sets to generate. And if we generate one that's not very insightful, that's not very clever.
You know, I suppose you can consider a human tumor as like the ultimate bioreactor of diversity, right? And ultimately what we see in clinic is probably the state that we need to learn about the most. And so how far, I mean, in your company, how do you think, how far are we away from that true, meaningful, real-world clinical context? And I don't want to preview my talk, but I'm going to talk a little bit about that. But I'd love to hear your thoughts from a Genentech giant company perspective. How far away do you think we are from that?
So it depends what we're trying to predict, right? Can we? So are you asking about, can we predict, see what will happen with a particular intervention? Is that basically the question? Like if I've seen this number of real tumors,
sure.
And
ultimately, that's where we want to get to. But I'm just thinking about in the lab in the loop context. Are we far from the real patient context? Are we able to approximate that with the lab in the loop context? And to what extent can we refine the lab in the loop context to get closer to
so
closing that gap and making that 1% into 5%, let's say.
Yeah, so taking samples and then predicting from a patient, I want to do this assay, and then it's going to... it's not really my area, so I don't want to speculate too much, if I'm honest. I'm happy to speculate in a quieter setting. But this one, there's too many people to know that what I say is absolute garbage. So I'll speculate with you in the break.
All right. Well, why don't we leave it at that? That was fantastic. Thanks, Sarah.